Hello, my name is Scott Acton, and welcome to Charlottesville, Virginia. I wish I were in San Diego right now with all my colleagues and friends at the SPIE meeting, but instead I'll just have to dream about those tacos in La Jolla at the taco stand, hanging out at Ballast Point and Miramar, walking through that beautiful Hotel del Coronado. So instead, I'm joining you from home, and I'm going to talk to you about image analysis and neuroscience and a little bit about the National Science Foundation. So let me share a screen. My talk today is called Image Analysis for Neuroscience. And I'm also going to talk about relevant signal, image, and data processing programs at the NSF that might help you out as a researcher in the long run. Again, thank you to the SPIE. Thank you to the Optics and Photonics Program Committee, and also to Khan Iftikharuddin for inviting me. My name's Scott Acton. I'm at the University of Virginia, and for the time being, I am a program manager at the Computer and Information Science and Engineering Directorate at the National Science Foundation. So let me tell you a little bit about myself. I was actually born in Southern California, which is why I'm missing the conference so much. My family relocated to New York. That didn't suit us very well. So we ended up settling and essentially growing up in Northern Virginia, where I went to Oakton High School. I also went to undergrad at Virginia Tech in electrical engineering, and then took a very obnoxious red Camaro and drove it to Texas, where I met up with Al Bovic at the University of Texas and did a master's and PhD. I'm very grateful for Motorola for supporting my PhD work. And I worked there for a bit before I ended up at Oklahoma State University in Stillwater, where I began my academic career. A few years later, I went cross country again, this time in a red truck, back to my home state of Virginia, where I've been a professor at the University of Virginia for almost 2 decades. I'm now about 100 miles away in Alexandria, Virginia, as a program manager at the National Science Foundation. I would love to have coffee with you in Alexandria once this whole thing ends, okay? So let's talk about neuroscience. Ramoni Cajal said at one point, as long as our brain is a mystery, the universe, the reflection of the structure of the brain, will also be a mystery. So this talk, at least the beginning of it, is about 4 questions that are posed by my 4 collaborators in this work. Essentially, these are 4 questions, maybe out of 400,000, that we need to answer before unraveling the mystery of the brain. The first question is about Alzheimer's. Does this newly discovered lymphatic system in the brain have anything to do with degeneration in the brain? We think so. The second question regards what I think is an open question in image analysis, which is, how do we quantify morphological differences between 2 neurons? And that's going to help us also analyze brain degeneration. Third, I have a question from my friend Taji Harris, who says, what do glia do? And we're going to look specifically at what glia do in a homeostatic or equilibrium state. And finally, and a little bit more generally, my colleague Andreas Gallman asked the question, how do we image and analyze living cells at resolutions below the diffraction limit? Well, I can help him with the second part of that question, just not the first. He's going to have to do the first part. So let's look at the first question first, which comes from Yoni Kipnis, a well-known researcher in neuroscience and in immunology. This is true. Just 5 years ago, scientists believed there was no lymphatic system in the brain. We're very familiar with lymph, aren't we? But we didn't know it was in the brain. And researchers at the University of Virginia, at around 2015, discovered this lymphatic system. And the question that remains is, could this system that gets rid of waste affect degeneration of the brain? This is something that affects 1 in 3 seniors in the United States of America. Well, first, we have to be able to image and to analyze images of lymph. And we've been working on this for a number of years. And these are results from my graduate student, Nazia. You can see an original image on the left, ground truth done by a technician in the middle, and our lymphi results on the right, which is anecdotal, but we're showing that we can extract these lymph vessels. And essentially, this is a standard level set approach with a twist. And the twist is that we're going to use a matting method that's used in the study of human optics to remove background vessels. We're going to use a matting method to classify pixels as either foreground or background. And when we're constructing our level set, which defines our segmentation, we're going to push the background pixels down with the negative sign and the foreground pixels up. And then we're going to do what we call a tubularity flow field segmentation that's detailed in our paper. And we're also expanding this to a convolutional neural networks solution that's going to be debuted in November of 2020 at the Asilomar conference. So what can we do once we get the morphology of this lymph structure? Well, we can study its function as well. We can look at diffusion and advection of waste through the lymph. Now, you can think of throwing a handful of flour that you use to make bread into a river. Part of it's going to spread out due to diffusion, which is our left part of this differential equation, and part of it is going to go with the flow, which is the advection, which is the right portion of this differential equation. So essentially, we've used this model to look at flow through lymph in the brain. And the cool thing is that we were able, if you look at the graph, to see normal waste removal in light blue and abnormal waste removal in dark blue, where we have ablated the lymph system. And we've shown that it's a significant difference. And this information and this approach was used by the Kipnis lab to actually make ties between Alzheimer's and the lymphatic system. This was lauded by Nature Medicine as a notable advance in the category of aging, and was also noted by Forbes as groundbreaking research that furthered the study of Alzheimer's, And you can see the very beautiful cover. I wish SPIE and IEEE had covers like this. We just can't match nature and their artists, I suppose. So this was exciting to be a part of this research. And this is one step in answering that first question. So what's the second question? The second question regards morphology of neurons. And Barry Condren, my fellow UVA professor, and also my teacher in biology, I took his neurobiology class. He says I flunked it. He asks, how do we quantify morphological differences between two neurons? Let's look at that. We've been working on this for a bit. And we take a little bit of a different approach. Instead of making a graph of a neuron and thinking about the graph in terms of edges, we think about the graph in terms of paths. And so we think of the neuron, in this case, it's a simplified three-path neuron. That's essentially the union of three paths. Each one of these paths has a position in three space. It also has some other features that I want to note, including concurrence. At a given point, one point is used by several paths. That's the concurrence of that point in a path. There's also hierarchy. You can think of how many bifurcations are above a given point going towards the root of this graph. And this is a tree. And that is the hierarchy. So at every point in a path, we have position, we have concurrence, and we have hierarchy. And there's other features, but I won't complicate the discussion for right now. When we think about comparing positions, we can think about procrustes and trying to rotate and scale and make the best fit between two neurons. But that actually doesn't work out from our perspective. If you take a Euclidean approach, say for making the one-legged man into the two-legged man, shown in red, you get a bunch of intermediate steps that don't make sense. Now, if you take an elastic approach to changing curves from the one-legged man in blue to the two-legged man in blue, there are intermediate steps that seem to make sense. Now, this is because of the shape manifold. If we can put our curves on what we call a square root velocity shape manifold, simply by taking the sample points, looking at their tangents, and making unit tangent vectors at the sample points, we can then measure differences between curves that form an elastic deformation from point A to point B, in this case from curve A to curve B. We use this in our neuron comparison. So here's our basic setup. Let's say you have two neurons. We're going to extract the paths. We're going to extract these features, concurrence, hierarchy, position, and we're going to auction off paths between the two neurons based on a Hungarian algorithm. Once we have this correspondence, we can form a cost function. You can see the cost function in the lower left. It has a penalty for deviating in concurrence. It has a penalty for deviating in location, which is elastic in this case, and has essentially lessened importance for greater hierarchy. You can see the term in the denominator. Now we're going to be able to form a distance between two neurons by summing these distances between paths that are corresponding, and then we can classify neurons. We can say, hey, this is an interneuron, or this is a Purkinje neuron. Now, what's more, if you look at the right side of the slide, we can take intermediate steps in between two neurons and show how they deform elastically. And this is going to be important in modeling degeneration of neurons, which is something that we're doing. Let me show you an example of this degeneration. In the upper left, you see a segmented neuron that has 304 paths. It degenerates over time to a neuron that has only 6 active paths in the lower left. The steps along the way can be modeled by our process that we recently published in Neuroinformatics this year. So to look at the details, I encourage you to check out this article that was led by Tamal Badabal. Now let's look at the third question. In the third question, Dr. Harris asks, what do glia do? Well, first of all, what are glia? Well, glia are more abundant in the human brain than neurons. I don't know if you knew that. These are resident immune cells that play a significant role during development. We know a lot about their role in development. We know less about their role in homeostasis. Now, these glia that you see in green, stained in green, have the large soma, the cell body, and you have these tentacles, these processes that are extending from the soma. These processes are thought to surveil the tissue for infection, for cell death, for debris, etc. And here you're seeing two videos in time of resting, so-called resting glia on the left and activated glia on the right. We would love to analyze these images and extract bioinformatics that tell us information about what the glia do in normal functioning brains, and in the case of infection. So just a little bit about the math of segmentation. I won't bore you. You can look at the paper from ISIP 2018 if you so choose, but it's basically a set of coupled level sets. The level sets have an energy function that you want to minimize that has a term that smooths. The regularizer, it has a term that evolves, that expands to capture, in one case, the filamentous structures of the processes, and in the other case, the sort of blobby structure of the soma. It makes sure that they don't overlap, that's the repel force, and it also makes sure to repair little broken pieces where our imaging is poor, that's the attraction force. Altogether, we can make a nice segmentation. You can see some examples. On the left, we have a glia from a healthy mouse. On the right, you have a glia that is infected with Toxoplasma gondii. And you can see differences in morphology here. You can see differences in ramification, where ramification essentially is the filamentary nature of these processes. So what can we do if we actually want to do an experiment and do some analysis on these glia? Well, on the left, you see an original image. And of course, we can only show you 2D on this 2D world. Our world is becoming increasingly more 2D as we use Zoom instead of real life interaction. And on the right, you can see essentially what we've done. What Tiffany Lee has done is she has segmented these cells and converted them into skeletons. These skeletons, we can get all sorts of cool information about the skeletons, the velocity of the processes, the length of them, how far they extend, how fast they're moving, how fast they're interchanging information. And we can do things like form graphs. One graph could be ramification, which essentially measures how different is the cell from just a standard sphere. And as you can see, healthy glia are more ramified than infected glia. And healthy glia also look at more volume in their surveillance than do infected glia. That's the graph on the right. So, and there's dozens of graphs we can show you, but the point is we can give researchers in biology and in medicine this information and do so repeatedly. Okay, that brings us to the fourth question of my research portion of my talk. And this is given by Andreas Gallman. And he says, how can we look at things below the diffraction limit and look at things that are living? And this, the reason it's more general than what we've talked about so far, this might deal with the brain, it might deal with other parts of the anatomy. And we all know at SPIE about the diffraction limit. And we know that we have tools below the diffraction limit, including electron microscopy, EM. But the process that we have to use to prepare specimens for electron microscopy basically precludes our looking at them as living, interacting organisms. So recently, there's been tools, single molecule fluorescence microscopy that have been developed that essentially move down in the resolution scale from conventional confocal microscopy, for example. And my colleague Andreas, he's looking at some interesting microscopic technology, specifically lattice light sheet microscopy, and he has his own version of this microscope that has taken months and years to develop. We get input from this microscope and we put the input in a neural network. You're probably familiar with UNet. We use a modified version of UNet to essentially take a first stab at segmentation. The UNet gives us numbers that represent essentially the probability that it's an interior of a cell, a boundary of the cell, or a background pixel. And we can't stop there because, as you'll see, there's broken cells and there's cells that are merged. We have under and over segmentation. And that's where Viva comes in. And that's where my student, Jia Wang, has done some really nice work. We have a post-processing method that we call L-cuts, and this gives us a final output. You can see her work along with the work of the Gallman lab already on the bioRxiv, and you should look up with keyword BCM3D. Okay, now L-cuts, how does it work? Well, we get these under-segmented and over-segmented regions, excuse me, and what we want to do is we form point clouds. And from these point clouds of what we think is a cell segment, we then compute medial axes. From these medial axes, we view connected portions as graphs. Then we find linear portions of that graph and successively split and merge until we're finished with providing the final segmentation. From these spines or axes, we can recreate the biofilm as you see on the right. A biofilm is essentially a colony of bacteria, in this case, Shionella in the example that I just showed you. And we're trying to look at these dense biofilms and count the cells and figure out how the cells move, how they get nutrients, how they deal with waste, this sort of thing. Now, here's an example raw image on the upper left from our lattice light sheet microscope. And in this case, we have ground truth, meaning that a technician actually marked each cell manually. What a laborious process. Now, if we just use the neural network, the convolutional neural network, we would get results as shown in lower left. But we have many cells that are stuck together because it's so dense, because the signal to background ratio is so low. And we also have cells that are split in the initial segmentation. Our L-cuts method that essentially uses intelligently the morphology of the cells helps put these pieces back together and helps split pieces that don't belong together apart. Now, one of the keys to our success is that we've developed a simulator. We can simulate images of cells as imaged by lattice light sheet. Now, these images give us a new database for which or with which we can do data augmentation in our neural network. They also give us truth because we would like to know, are we doing it right or are we doing it wrong, incorrectly, in terms of segmentation, in terms of counting. And now we can do that with this simulator. Then we can also test on real data to make sure that we're not essentially deceiving ourselves with an easy simulation. This is a result on the right of the neural network plus L-cuts post-processing and one of the simulated images on the left. As you can see, it's a difficult segmentation problem, and that's why we love it. Okay, so my colleague, Barry Condren, and you saw his question and some of his neuron work earlier in the talk, he likes to say, and maybe we're on mile 2.5 because he said this a year ago, we're on the second mile of a 26-mile marathon. And there are tons of questions to be asked and tons of answers to be had. And it's my belief that it's only through collaboration between biologists and engineers, computer scientists. It's only through this collaboration that we can have success. And our success will be a little bit at a time, I think. And this is one of the big problems that's out there in science and engineering is how does the brain work? So thanks for listening to that portion. Now I'd like to talk about the National Science Foundation. And I'd like to talk about programs that might be relevant to you, a person working in signal, image, and data processing. So I'm in a cluster called Communications and Information Foundations, CIF. That cluster contains 3 program managers, Phil, Armand, and myself. We're one of the most accessible clusters in the foundation, we'd like to thank, and we would love to talk to you at NSF. We'd love to answer your questions by email. I can be reached at sacton@nsf.gov. We fund a variety of programs across the country, apparently not in Wyoming and South Dakota, but we're growing in our support of EPSCoR states, and we're also growing in our support of underrepresented minorities, including women, in our field of computer science and electrical engineering. Some of our topics, these are the main topics: signal and image processing, which is what we're talking about today. Machine learning and optimization, that's a big one. Probably half of our proposals or more have some element of machine learning in them. Information theory, coding, communications, and networking. We would like to believe that we've had an impact on the world. We'd like to believe that our programs and our researchers have made applications that really benefit you. One such application is speeding up MRI scans. If you've ever sat in an MRI machine, you know it can be uncomfortable, loud, and not the best experience. Researchers at Rice and other universities, through NSF funding and other funding, were able to dramatically improve these times. And companies like Siemens adopted these technologies, and that's a big success story for signal and image processing and data processing. Whether you know it or not, you probably even today used some of the technology funded by the National Science Foundation in repair codes. These repair codes are used to reliably store information on cloud systems. So if you use Google today or Facebook or Microsoft, maybe these were codes that were actually pioneered by researchers with NSF funding. And we're proud of that, and we're proud of our researchers that did the work. We even have a movie. We helped fund a movie about Claude Shannon. It's called The Bit Player. I watched it a few weeks ago. It's beautiful. It's a wonderful movie. You can find it on Vimeo, I think for free. And it's a quite enjoyable movie to watch with your kids and to learn about a hero of information theory, Claude Shannon. So at the NSF, I work in my normal program, but I also work in Smart and Connected Health. I really enjoy this program. It's cross-cutting. You can check it out on the web. We have lots of great SPIE researchers that are in our funded programs. The projects are integrated between medical personnel and computer scientists and engineers. They can be pretty beefy in terms of money at $1.2 million. NIH helps fund this, as well as the NSF. And it's really use-inspired basic research. This research it's at the intersection of medicine and science and engineering, but we really want basic research that has as its inspiration medicine. And you can contact Wendy, you can contact me about this, but this quadrant explains what we really want. We want something that's high in the quest for basic understanding first, and also is application inspired second. We don't want one or the other. And we certainly don't want one that lacks both. So we're looking for sort of Louis Pasteur. And what an appropriate time to mention a person like Pasteur that dealt with disease in an earlier time in the 20th century, and now we're facing a crisis now in the 21st century. Speaking of that crisis, we have funded in our directorate a few hundred small initiatives on COVID-19. These small initiatives are $100,000 to $200,000 grants where people with brilliant ideas thought they could make an impact in 6 to 9 months. We're starting to see the flowers bloom from this program, and we're expecting, and you should be expecting, new opportunities on the pandemic in terms of researching and helping out medical research in this area shortly from the NSF. We believe that imaging and photonics, those are key SPIE areas, are also key to addressing this virus and this pandemic. I know you've been affected by it. You can tell that we all have, since we're doing this by teleconference, and we're hoping that brilliant minds such as yours can be lent to this, to the solution, which will probably require many facets and many solutions in the near future. I'm also helping out with robotics at the NSF. There's a new program on foundational research in robotics, which should be exciting to this data processing community. You can look it up on the web. You can ask me questions. You can ask Dave Miller questions about it. He's a rotator from Oklahoma and is in charge of the NRI program at this point. Image processing vision, obviously part of robotics. And also obvious is the fact that we all work in an area that's been profoundly affected by artificial intelligence, specifically machine learning. You've heard about our institutes. We're going through a second round. There are going to be some exciting topics that are going to be relevant to the SPIE community. I encourage you to start forming teams and start thinking about the next round of these huge institutes that are directing the country's direction in machine intelligence. You can always find information about engineering and how it relates to your work. One program I'd ask you to look at is communication, circuits, and sensing systems within the engineering directorate. This is a program that does a lot of imaging, that does a lot of signal processing, and is a program that we think about as a sister program to our program in size. So please contact Dr. Lin if you have questions on that program. There's plenty of other opportunities. You might be surprised to find out that I've been funded by education and biology. And I'm not an expert in either, but I've used signal, image, and data processing to contribute to these fields. Geosciences is another area ripe for innovation, as well as neuroscience. You can contact Ken Wang. And cyber-physical systems is something that has been a really fruitful area for researchers at the SPIE conference and in terms of image and signal processing. So I'd like to conclude. Innovations in signal, image, and data processing are really at the core of recent innovation and technical technical revolution that we've experienced, excuse me. So it's the key, as I've been telling you, the key to reverse engineer the brain. It's the foundation of devices like the smartphone, for better or for worse. Signal and image processing, really the key to autonomous vehicles and communications to V2V and V2X. I would contend that without knowing signal, image, and data processing, you can't know neural networks. How can you know convolution without knowing the basics of signal processing? And so, as signal processing people, we form the theoretical underpinnings of the neural network community. We also contribute to the Internet of Things, and I'm excited about putting some rigor in data science. I'm excited about your work, and I really appreciate you taking the time to listen to me. Again, my name is Scott Acton. I'm at the National Science Foundation in Alexandria. I'm also at the University of Virginia in Charlottesville, and now I will take your questions. Thank you very much.